All right, well, let's get underway. I guess it's 3:31 by my clock. Welcome everybody to the Botany and Plant Pathology Departmental Seminar, co-sponsored by the Center for Plant Biology. It's my great pleasure today and a distinct privilege to be able to introduce Dr. Federica Brandizzi from Michigan State University today. Federica is a professor in the Department of Plant Biology, but she's also a member, and it's probably her key role is as a member of the Michigan State DOE Plant Research Lab, where she's been for the last decade and a half, or a little bit more actually. She reminded me, and I should know this because we participated at the EFRC level, she's also the science director for the DOE-funded ERC called the Great Lakes Bioenergy Research Center, which is now in its 10th year and up for renewal for a third round. And we wish them luck on continued success with that effort. And a few of you in the audience may know Federica from her role as a senior editor at the Plant Journal, where she's done a lot of incredible hard work over the years. Federica's had a long and distinguished career at Michigan State and has been recognized for her numerous scholarly and creative achievements. And her achievements span a spectrum from fundamental to applied in plant science. Among her many honors, in 2015, she was named Michigan State Foundation Endowed Professorship, a named professorship. In 2018, she was elected as a fellow of the AAAS. 2 years ago, Michigan State recognized her for some of her applied work as the 2020 Michigan State Innovator of the Year. And just last year, she was recognized and named as a distinguished professor at Michigan State. So awesome set of career credentials and recognitions for her amazing accomplishment. Federica, for those of you that don't know her work, and hopefully after today you know very well about her work, Federica has really been a pioneer in combining live cell imaging at a really high spatial and temporal resolution with molecular genetic analyses to be able to dissect the endomembrane system of plants. And in particular, she has a focus on the secretory pathway, the forward pathway for cell wall deposition, biomass creation, et cetera. Test the microphone still on, buddy. Federica has almost 200 publications to date. Most of these are in top tier journals. Many of them are incredibly highly cited. And I was going to scroll through and refamiliarize myself with some of her work. And I was just astounded to see the number of publications, especially in the last 5 years. It's been an incredibly prolific run for her in her lab. Today, without taking more time, Federica is going to be telling us about the remarkable gymnastics of the plant ER network, and its role in plant growth and development, as well as in plant response to stresses. So please take it away, Federica, and welcome to Purdue, virtually.
Okay. Thank you so much, Chris. Thank you for inviting me. It's really an honor. And as I was saying earlier, it's just fantastic to talk with colleagues, talk about science. And after this hiatus of COVID, having human interactions about science, although virtual, it's been a fantastic day. So I'm very grateful. Thank you so much. It's been really my honor. Chris has made a list of accomplishments, but in reality, it would have anything would have not possible without people that work in the lab, people that do the hard work, and that I take credit for, really. So some of these people are here listed, and today I'm going to show you some of their work. The topic of the talk, like Chris mentioned, would be the endoplasmic reticulum, which is an amazing organelle of all eukaryotic cells and is responsible for the synthesis of 1/3 of the proteome, the cellular proteome. And as such, it builds the cell, but also allows the cell to communicate with the external environment. Today, I'm going to talk to you about how the endoplasmic reticulum, I'm going to call it ER for short, interacts with other organelles, but also, surprisingly, science is taking us to different level of interactions. We're going to see the interactions at the level of other pathways that go well beyond secretion. So endoplasmic reticulum is the core of the secretory pathway. The secretory pathway is a set of organelles that communicate with each other to transport the proteins that are synthesized at the level of the endoplasmic reticulum through the Golgi apparatus. And from the Golgi apparatus, proteins and membranes that allowed such transport are then transported to intermediate compartments that are called endosomes. And from here, proteins can either be delivered to the vacuole or to the plasma membrane. Counteracting this enormous flow of proteins and membranes towards the plasma membrane is a process called endocytosis, which allows, again, membrane and proteins and external content external to the cell to be integrated back into the cell. And so this is how really the secretory pathway is a conduit with the external environment, both for the in exocytosis, so forward transport, and backwards via endocytosis. And so understanding how the secretory pathway works is fundamental in cell biology, but also has many applications in the field, because we can potentiate this pathway to produce more proteins, more membranes, and as such making plants that are bigger and more resistant to a number of stress. And so there are a number of beneficial returns to the human population to understand better how this pathway works. Our focus is the endoplasmic reticulum, and really is really is the protagonist of a big show here. What I'm showing in this movie is the cortical region of a cell that's been transformed with a marker that is targeted to the endoplasmic reticulum. What you see here, therefore, is just the cortical endoplasmic reticulum. And you can appreciate that this organelle is made of tubules that are continually retracted and reforming, but also cistern, these more enlarged parts of the organelle. Now the video is undergoing quite a bit of a speed, but if you go to the confocal microscope, with which we can take this kind of movies, you will be just fascinated by this enormous movement of membrane that just relates to just one organelle. So what you're seeing here is a continuous of membranes. It's not more numerous organelles put together. It's just one. As such, because it's so pervasive into the cell, the endoplasmic reticulum connects with many other organelles. And many years ago, the late Professor Chrysos really established with microscopy the foundation in understanding how endoplasmic reticulum interacts with other organelles. And what I'm showing you here is a movie of a cell that expresses in green a NIA marker, and then in magenta, the Golgi marker. The Golgi in plant cells, different from mammalian cells, is dispersed in these little stacks that you see here. And what Chris did, he used optical laser tweezers to grab one Golgi stack and then move it around in the cell. In this cell has been treated with some inhibitors of the cytoskeleton to block the ear from moving, so that there would not be complications from the movement of the ER. But what he's doing here, Chris is just taking one organelle, one Golgi, and then moving it. And as you can see, the endoplasmic reticulum is actually following the Golgi. So these two organelles are attached. We do not know how they're attached. We know about the machinery. I'm going to run this movie again because it's just fascinating. We know the machinery that functionally connect these two organelles. The machinery is called COP2 and COP1 for protein transport. but we do not know what really attachment points between these two organelles can be. Now, the endoplasmic reticulum is also attached to other organelles, and I can show you some evidence in this respect. For example, the mitochondria that you see here in red are traveling along the endoplasmic reticulum, which is in green. And there is a close relationship in plant cells. Again, we only know about one protein, it's called myro, that the group of Imogen Sparks has been working on, and maybe the connection between the mitochondria and the ER. The ER also provides membranes for the biogenesis of peroxisomes. And again, you can see how peroxisomes in red are in close association with the ER. And then the Golgi stacks. Again, you can see how the Golgi are moving on the endoplasmic reticulum, although we're not using here optical laser tweezers to move the Golgi around. So there is a lot of movement in these cells. The endoplasmic reticulum is moving along, so are the organelles. So we wanted to know a bit more about this relationship between the endoplasmic reticulum and the other organelles. And so we start looking, we start using a mutant that is called RHD3, which stands for root hair defective 3, which was identified early in the 90s by the group of Scheffelbein. And this mutant has a very short root hair growth. But it has also a fascinating phenotype of the endoplasmic reticulum. Essentially, the endoplasmic reticulum of fully expanded cells in mutant, in the RHD3 mutant, does not look like the beautiful network that you will find in a wild-type cell. The tubules are collapsed in these long strands of unbranched tubules, and the fusion of tubules with other tubules is compromised. Indeed, RHD3, we discovered later, is a fusogen, so is a protein that is responsible for the homotypic fusion of the membranes of the endoplasmic reticulum. So the loss of RHD3 causes this fascinating phenotype. By studying RHD3, we first noticed that when cells are expanding, the endoplasmic reticulum changes shape. So let me back up a second here. We followed cotyledons throughout the cell expansion. The group from Danzimansky had nicely shown how cells expand and how we can time the expansion of these modified leaves in Arabidopsis. And so we used that time that was identified as essential stages of rapidly expansion and then full expansion of the cells to follow the morphology of the endoplasmic reticulum. And so we found that very early in development, the endoplasmic reticulum does not look like the mature endoplasmic reticulum. It looks like really a sheet of membrane that then evolves in this beautiful reticulated network that we're all familiar with. In RHD3, we have a similar situation in Colombia, in wild type, at very early stages of development when tubules are not there yet. But as soon as tubules should start developing, then we have this large collapse of the tubule themselves, indicating that there is a progression maturation of the ER, and the RHD3 is really important for that. So we then measured the velocity of the movement of the endoplasmic reticulum, both in wild type and RHD3. And we found that as cells expand and form this beautiful network, then also the velocity of the endoplasmic reticulum is increased. However, in RHD3, such increase is very much reduced. So at this point, having this genetic tool raised the question for us, okay, if the endoplasmic reticulum connects and contacts with other organelles, is it possible that it influences also the movement of these other organelles? And so we used, to address this question, we used microscopy And we used plants that expressed a Golgi marker, mitochondria marker, or peroxisomal markers, and it looked at cells that were very early in development, so the cells were not expanded, or in fully expanded cells. And then we took images through in time-lapse microscopy, and each image was assigned a different pseudo color. And so when these then images are piled up all together, you will notice that if organelles have not moved, so they've been stationary, you would see a predominance of the color white, which will be the last color in the series of the time series. Otherwise, you will see a rainbow of colors. And this is really nicely highlighted here in wild type, whereby 3 days of from germination, when the cells are not expanded, the organelles like the Golgi have not moved much. However, in 12 days, when the movement of the endoplasmic reticulum is really elevated, then also the Golgi movement is elevated. Same thing happens for the mitochondria and for the peroxisomes. But then we asked the question, okay, what would happen in RHD3, where the movement of the endoplasmic reticulum is reduced to a specific mutation that affects the development and the movement of RHD3. And so we noticed that in cells that should be completely fluid in terms of movement of the Golgi, mitochondria, and peroxisomes, now the color white is predominant, indicating that these organelles are not as motile as they would be in wild type. And so this really is telling us that the endoplasmic reticulum has a dominant effect on the movement of these organelles, and possibly this happens through connections. And another example that I would like to bring forward is a collaboration that we had with Marisa Otegi at Wisconsin-Madison, where we showed that also endosomes that have a close association with the endoplasmic reticulum. Marisa did this beautiful 3D electron tomography to reconstruct the endoplasmic reticulum and endosomes association is demonstrated that is very close. And I'm sorry, the movie is not working here, but you would have seen the endoplasmic reticulum and the endosomes moving together. Now, when we look at markers of the endosomes in wild type and in the RHD3 mutant, again, we see that in fully expanded cells, whereby the endosomes in wild type move all around. Now in RHD3, the movement is really restricted. So fascinatingly, the endoplasmic reticulum is not shy to interact also with, if you want, very large organelles. It takes it on the big boys, like the chloroplast. And this is beautiful work shown by the group of Anderson that many years ago isolated protoplast expressing an EM marker and then visualizing the chloroplast with the autofluorescence of the chlorophylls. And what they did, they burst the chloroplast, and then with the protoplast, with optical laser tweezers, they pulled the chloroplast away, and you can see that the endoplasmic reticulum is following. So the endoplasmic reticulum is also attached to these organelles. And what they did that I found fascinating, they even tried to shake off the chloroplast, but the endoplasmic reticulum, you can see in green, is really attached. There's nothing to do. These are attached. So the point, though, now that I think is really important to scientists is finding how do these organelles interact with each other. We know there is a physical interaction, and group measurements from the Anderson group indicated that the strength of the interaction, for example, between the endoplasmic reticulum and the chloroplast is such that can be only supported by a proteinaceous entity. So what are these proteins? So just in our small world, we are attempting to identify these proteins, and I would like just to give you an idea of how we go about doing this. We just use brute force microscopy. So we were fortunate to participated in a collaboration with Paul Dupree in Cambridge. And at that time, Paul had established the first draft of the Arabidopsis organelle proteome. And they had isolated organelles and then started understanding or characterizing proteins that were associated putatively with these, with the organelles that they isolated. And we were involved in tagging some of these proteins to test where they would be localized and confirming the proteomics results. And so out of these 182 proteins, I think we tagged most of them with fluorescent proteins. And we're now coming across proteins like LURE1 and LURE2 that I'm describing here, that you can see that they are localized at the endoplasmic reticulum, but they also form this beautiful half moon shape in the case of LURE1, which surrounds the chloroplast, and LURE2 forms these dot structures around the chloroplast. So this is just to give you an idea on how we can go about and start identifying machinery that hopefully we'll be able to characterize in the future. Now, the point though is, okay, maybe we can identify the machinery, but what is the purpose of the interaction? And sometimes it's not that immediate, right? Why would the endoplasmic reticulum interact with other organelles? In the chloroplast, for the chloroplast at least, the reason is there in the sense that these plastid membrane association contacts, they are called plums, have been invoked as being very important for the transport of lipids between the endoplasmic reticulum and the chloroplast. And this is an essential process that is fundamental for the life of the cell, for the production of lipids, both that reside in the chloroplast, but also lipids for the entire cell. So having these points of contacts makes sense for the endoplasmic reticulum and the chloroplast. But what about the other organelles? I don't have an answer for all the organelles, but what we've been studying in my lab is the interaction of the endoplasmic reticulum with the plasma membrane. So the endoplasmic reticulum is anchored to the plasma membrane, a bit like a spider web hanging from the ceiling. And this is a movie, this is in seconds, it's going really fast, but you can see that despite the high movement of the ER tubules, of the cisternae, of the strands, there are points that never move. And these points are highlighted by a protein that is called BAP27. BAP27 was characterized initially by the group of Patrick Hussey, which showed that just a little bit before us, that the BAP27 is localized at the endoplasmic reticulum and is at these points of contacts with the plasma membrane. And BAP27 interacts with an actin binding protein, NET3C, but also with microtubules. And so these sites that they called EPCS, or ER plasma membrane contact sites, are really hubs for cytoskeleton and the endoplasmic reticulum to anchor these organelles to the membrane. We wanted to expand on this fundamental work that Patrick Hussey had put forward, and we wanted to understand what is the role of these contact sites between the endoplasmic reticulum and the plasma membrane. So we first did a pull down of the BAP27 proteins. So Arabidopsis has 5 of them. And we're finding that BAP27.1 and BAP27.3 are the major isoforms. And we pull down BAP27 proteins to identify, to realize that they're actually interacting with clathrin. Clathrin is a very important protein that forms a coat around endocytotic vesicles, but also is situated at the post-Golgi compartments like the trans-Golgi network. Both the clathrin and the trans-Golgi network are very important in endocytosis in plant cells. So we could confirm the pull down in vivo using these two hybrid between the VAP27 proteins and also with FRET analysis. I'm not going too much in detail here, but what I would like you to see is that when we use a functional fluorescent protein fusion to clathrin in cells in co-expression with VAP27 fluorescent fusion, we saw that the clathrin structures were always in association with the plasma membrane contact sites as well as with the trans-Golgi network. So VAP27 proteins seem to be really a hub that recruits not only cytoskeleton, but also clathrin to these plasma membrane contact sites. So then we wanted to test whether there was a functional connection between VAP27 and clathrin. And so we generated a double mutant of VAP27 1 and 3 and looked for processes for endocytosis efficiency. And this is something that can be easily followed with internalization of a dye that is called FM464. FM464 labels the plasma membrane. We are now in roots, and after a few minutes, the marker is endocytoted into these structures, which are endosomes and trans-Golgi network. And so we looked at the internalization of the dye in the VAP27 double mutant. And you can see here that the internalization of the marker is much reduced compared to a parental line control, so to a wild type. We quantified all this, but the bottom line is that this was an indication that endocytosis is reduced in VAP27 mutant. So then when endocytosis happens in cells, there is the formation of the endocytotic vesicles at the plasma membrane. So these assemble, the cargo is with the vesicle is brought into the cell, and then due to GTP hydrolysis, the clathrin is uncoated from the vesicle. And this leads to a so-called blinking of the clathrin-coated vesicles. And if we're lucky, this movie will work, but it's very fast. But you should be able to see that vesicles appear and disappear. And this is the process of blinking. So we quantified this feature both in wild type and in the VAP27 double mutant. And we see that the blinking happens within 15 seconds in the wild type. So we have the formation of the vesicle and then its disappearance, but the process is much reduced in the mutant, at least in terms of time. There is kind of a delay in the formation of these vesicles and in their blinking. The clathrin-coated vesicles also wiggle about before detachment from the plasma membrane, and this is called lateral movement. And we've seen that in the mutant, the vesicles are hanging at the plasma membrane for quite some time, while in wild type, this movement, this lateral movement is let's say, is normal. It happens. Something that does not happen as fast in the mutant. So we then looked at another aspect. So if you consider how could an ER protein be associated with the plasma membrane, one answer could be, well, we show that clathrin, that VAP27 associated with clathrin, so that could be an anchor. But we also know that clathrin is assembled and then disassembled off the plasma membrane. So there must be something else. And we couldn't find anything that was convincing in our pull down that could form an anchor with VAP27 at the plasma membrane. So we thought that maybe it was a lipid anchor. And so we did a lipid blot analysis where we took the cytosolic domain of VAP27 and expressed them in a recombinant manner, and then did a lipid overlay assay. And you can see the results here that it appears that the VAP27 proteins interact with PIP lipids that are enriched in endocytotic membranes. So these data lead us to a model whereby the VAP27 proteins that you see here in red, that are anchored to the endoplasmic reticulum, recruit clathrin, and they can interact with the plasma membrane thanks to an interaction with PIP lipids. And these VAP27 proteins are really important for the recruitment of clathrin. And if there is not enough in the pool of VAP27 proteins, then you have a defect in endocytosis, most likely because you don't have efficient recruitment of the machineries needed for the formation and evolution of the clathrin vesicles. So I was particularly fascinated by this study that I wanted to share with you because I think it brings about a different model. So to have organelles interacting with other organelles, you need proteins, for example, on the ER, but not necessarily another protein on another membrane. And I was also very happy because we were contributing now to an understanding of what these points of contact of the endoplasmic reticulum with other membrane could be doing, what would be their function. So, so in this, I just want to summarize this part of the talk, and I hope I convince you that the ER is a pervasive organelle in the cell that can contact so many heterotypic membranes, and that we're identifying some of the machinery, and that the ER is such an amazing structure that is a propelling force for the movement and the life inside of the cell. And so I would like now to bring you to very unexpected results that we got in our lab that took us away from our typical organelle biologists that we are and took us to completely different pathways. So, so one centric view of the endoplasmic reticulum is the idea that its movement depends on actin. And so when we treat cells with latrunculin, which is an actin depolymerizing agent, and Chris can tell you much more than I can do on this drug and other, the endoplasmic reticulum loses the beautiful network-like shape and also its dynamic really entity. So actin is really important for the movement of this organelle. And we know that actin interacts with the ER also at the level of electron microscopy. So this is not just something that you can see in beautiful movies, and maybe an indirect effect of depletion of actin. Beautiful work from Andrew Stalling showed that the actin filaments are really in close connection with the ER. And so by following that study that I told you pre, that we were set to identify proteins that could connect organelles with DR with other structures, we came across a protein that is called CYP73. CYP73 has a transmembrane domain and then two actin binding domains, and is localized at the endoplasmic reticulum. You can see here in green. And these cells are also co-expressing an actin marker that you see in red. So in low level of expression, CYP73 decorates the endoplasmic reticulum. However, in high level of expression, then the endoplasmic reticulum assumes a shape that really resembles actin organization. So when we came across this protein, we got really interested. And we indeed demonstrated that CYP73 can interact with actin, it can pull it down. And so what we're proposing here is that CYP73 may be the anchor of the endoplasmic reticulum with actin. And so this is a study that we published a few years ago. And again, doesn't take us away from the ER-centric view of these, I don't want to call it expected interactions, but in a way it's expected the cytoskeleton can organize and move an organelle. But a question that comes to mind is, okay, so what does tell the cytoskeleton about moving organelles? So does the cytoskeleton always work in a constitutive manner to move organelles like the endoplasmic reticulum around, and then the endoplasmic reticulum moves the other organelles, or is there a superior force that can move membranes around? And so this is a story that I want to tell you that we stumbled across really by pure luck. So in our lab, we do lots of forward genetic screens using EMS mutagenized populations of Arabidopsis that express markers of the endoplasmic reticulum, the macula, and so on. And we look for defects of the organization of these organelles. And then we map the mutation and we identify really, I think, quite important proteins of either the ER or the cytoskeleton that can control the organization of the ER. But this time around, we came across something that we were not expecting at all. So we came across a mutant that we called EVA1. And EVA1 has this very stranded ER. You can see that the ER is almost collapsed. We're now looking at a leaf surface, just a section of a leaf surface of a plant that is expressing an ER marker. You can see at low magnification, there are all these beautiful stomata here. EVA1 has the stomata, of course, but then this really collapsed ER that you do not see in wild type at this level of magnification. If we increase the magnification, you can now see the beautiful ER in wild type, but in the EVA1 mutant, there are these strands of ER, collapsed ER. So we reason in this mutant, if the ER is collapsed, also the actin cytoskeleton will be collapsed. And indeed, by looking at an actin marker in EVA1, we found that the actin is assuming this hyperbundled configuration that you do not see in wild type. So we thought, okay, this is a mutant of the organization of either the endomembrane of the endoplasmic reticulum or the actin. But then when we mapped the mutant, to our biggest surprise, we found out that the EVA1 mutation is a missense mutation in IPMS1. IPMS1 is an enzyme that resides in chloroplast and is really important for the synthesis of leucine. Leucine, isoleucine, and valine are the three branch-chain amino acids that are indispensable for human nutrition and indispensable if you want to build up muscles in the gym. But importantly, the loss of IPMS1 it reduces the synthesis of leucine, and as such, the precursor is shunted towards the synthesis of valine and isoleucine. And indeed, in EVA1 and other alleles of IPMS1, we found that there was an elevated levels of leucine. So I expected everything, to come across everything, about the ER and the actin when you do this kind of forward genetic screen. But now we hit a mutant of a metabolic pathway in the chloroplast, and we see a big effect on the ER and actin. So what's going on? So Federica, who was involved in this study, tried all the possible drugs that we had in the lab to just, try to figure out, try to make a connection with pathways. And so in the lab, of course, we use oryzalin, which the polymerase is microtubules, and found that there was no resolution of the complex phenotype of EVA1. Latrunculin B, which the polymerase is actin, had some resolution of the phenotype. However, the most interesting drug was Wort Manning. And Wort Manning in recent years has been studied and used as an inhibitor of post-Golgi trafficking. But Fei was really clever and he started digging very old literature. And actually, he found that Wort Manning is an inhibitor for PI3 lipid kinase, but also of TOR. And so recently, Wort Manning has been used mainly as an inhibitor of PI3K lipid kinase, but actually, let's step back. This drug is also important for TOR. So it inhibits TOR. So TOR maybe doesn't need much introduction, but so that we are all on the same understanding here, TOR is an essential protein kinase that is conserved across all eukaryotes. And it integrates metabolic or nutrient signals. So essentially, it perceives nutrients like sugars. And then by determining that there is availability of nutrients, TOR then will promote growth and will suppress autophagy. Autophagy is the process of self-eating of the cell to survive in specialized situation like starvation. So TOR antagonizes autophagy. But TOR really is a major integrator of growth signals and is a pro-growth factor. So at this point, we were lucky because there are some drugs that are specific to TOR inhibition. And so we use two of them, AZD and Taurin-2. And we looked at the organization of the endomembranes in wild type and in EVA1 upon treatment of TOR inhibition. And so you can see that in wild type, inhibition of TOR leads to the accumulation of these structures, of these little punctae, which are autophagic bodies. In EVA1, we noticed that, yes, there was the formation of the autophagic bodies, but also that the typical structures of endoplasmic reticulum, but also of actin, also of vacuolar strands that I'm showing you here, essentially were made disappear by inhibition of TOR. So this is telling us that in EVA1, most likely TOR is hyperactive, and reduced activity of TOR eliminates the subcellular phenotype. We indeed confirm that TOR is hyperactive in EVA1 and in another allele of IPMS1 using known markers of TOR activation, like phosphorylation of the S6K subunit, ribosomal subunit, which is a target of TOR. And you can see that in EVA1 and an allele of IPMS, there is enhanced phosphorylation or abundance of phosphorylated S6K compared to wild type. Another hallmark of TOR activation is increased cell proliferation at the root tips, something that one can follow with EDU staining. And you can see that in alleles of IPMS, there is enhanced cell proliferation compared to wild type. So all this is telling us that essentially TOR is hyperactive in EVA1. Inhibition of TOR leads to suppression of the aberrant phenotype of the endomembranes. So at that point, we thought, okay, if you, maybe the phenotype is due to hyperaccumulation of isoleucine and valine that is due to the absence of a functional IPMS. And so we treated wild-type cells with branched chain amino acids and found that, in this case, I'm showing you actin cytoskeleton organization, there was a marked bundling of the actin. So wild-type is responding to BCAA, and the actin is bundled. And you can see this bundle. We draw a line to measure the pixels to identify areas of intense accumulation of fluorescence. And here's the graph where you can show that when there is the bundling, there is a peak in accumulation. So then we also looked at the other alleles of the biosynthetic pathway of the BCAA, and in IPMS allele, which accumulates, over-accumulates valine. Again, we have these spikes of actin aggregation, and also in the mutant ORM1, they over-accumulate isoleucine. So it really seems that this response to branched chain amino acids is what leads to the bundling of actin. And then, as we know from data I'm not showing you, also bundling of the endoplasmic reticulum, which is associated with actin. So at this point, we wanted to see what is the relationship between branched chain amino acids and TOR, because so far in plants, we only knew that TOR was responsive to sugars, but not to branched chain amino acids or other amino acids for that matter. And so we secured two mutants. One is an inducible, estradiol-inducible RNAi knockdown of TOR, which is called TOR-ES, and then RAPTOR1B. So let me step back a second here. In mammalian cells, there are two complexes of TOR. One is called TORC1, and the other is TORC2. Plants express homologous genes only for TORC1. We don't have the complex like SYN1 and Richter, typical of TORC2. And in plants, we also have a mutant of RAPTOR, is a knockout. So RAPTOR is part of the complex of TORC1 and is viable. So the fact that the absence of RAPTOR is viable, but we know that the absence of TOR, complete knockout of TOR, is lethal, means that there may be another complex in plants that is analogous to TORC2, but does not have the same proteins. But so let's now go back to the influence of branched chain amino acids on TOR activation. You can see that when we treated TOR-ES without estradiol, so these are just almost wild-type plants with branched chain amino acids, we have now a collapse of the actin cytoskeleton compared to plants that were not treated with branched chain amino acids. So it's really branched chain amino acids, again, is causing this collapse, as I showed you earlier. But now we activate disruption of TOR, and by treating cells with estradiol. And what you can see is that this formation of actin bundling does no longer happen. Then we looked into RAPTOR1B mutant and found that the bundling of actin in the presence of branched chain amino acids still happen. So what we can conclude here, which I think are major discoveries for the field of TOR, is that plant TOR actually responds to branched chain amino acids homeostasis. And this advanced the TOR field because, as I mentioned earlier, we only knew until our study that TOR was activated by sugar. So now we put branched chain amino acids. But we also have now an interesting phenotype at the subcellular level. There may be a TORC2 analogous complex that in case of hyperactivation leads to actin binding. And this then leads to the formation of YAS strands and transvacuolar strands that I showed you earlier. So for me, this study was fascinating because brings about a new level of complexity when we study cell biology. For a cell biologist like me that was very happy to work with organelles and cytoskeleton, now we have a major controller of the system that is TOR that leads to really organization of the cellular environment. But then, because in our lab we do so many other things besides studying only the morphology of the eye, we found another interesting connection that I want to show you with you. So I mentioned that the endoplasmic reticulum produces 1/3 of the cellular proteome. So it's a really busy machine. And by being very busy, it can also do a number of mistakes, which are usually caused by induced production of proteins. For example, when plants have to grow or they have to respond to pathogen attack, then the activity of the endoplasmic reticulum is stressed. And so really the endoplasmic reticulum assumes enhanced capacity to synthesize proteins. But the endoplasmic reticulum also has the charge to make sure that these proteins are fully folded before they are released to the environment, so that they are safe for the cell and for the surrounding environment. So monitoring the activity of the endoplasmic reticulum is a set of signaling pathways that is called UPR, or unfolded protein response. So there is always a level of UPR activation when cells are growing, but then when something kicks in, like sudden growth, attack to pathogen, the UPR kicks in, trying to help the endoplasmic reticulum synthesizing proteins properly. However, if the capacity of the UPR is overwhelmed and proteins, misfolded proteins accumulate to too high levels, then the cells initiates a process of pro-death. So the cell tries to commit suicide in order to prevent deployment of misfolded proteins in the environment. So the UPR is governed in plants by two arms, the IRE1 arm and the BZIP28 arm. So these are the major controllers. IRE1 is a fascinating protein of the endoplasmic reticulum. It's anchored to the endoplasmic reticulum by a transmembrane domain, and it has a kinase activity, but also a ribonuclease activity. And when it senses the accumulation of misfolded proteins in the lumen of the eye, it splices the mRNA of a transcription factor that is called BZIP60. BZIP60 goes to the nucleus and controls the expression of proteins that can mitigate the accumulation of misfolded proteins in the eye. BZIP28, again, can sense misfolded proteins in the ER, is translocated to the Golgi apparatus, where the transcription factor domain is released thanks to proteolytic cleavage, and then the transcription factor domain goes into the nucleus to, again, activate or modulate the expression of proteins that can control the accumulation of misfolded proteins in the eye. So many of the studies so far on these, on the activity of IRE1 and BZIP28 have been done with the use of drugs that cause protein misfolding in the endoplasmic reticulum. We also know from studies in our lab and many others that mutations in IRE1 or BZIP28 and also in other, let's say, minor modulator of the UPRs actually have a profound effect at physiological level. So without the implementation of drugs to perturb the system. And I want to bring your attention to IRE1. IRE1 has two functional isoforms in Arabidopsis, IRE1A and IRE1B. When both IRE1A and B are depleted in an IRE1 AB double mutant, then the plants have this very characteristic short root phenotype compared to wild type. So just the loss of IRA1 causes this incredible phenotype. So we wanted to look a bit more at this phenotype, and we started looking throughout development of plants. And we found that 5 days, 7, 10 days, 12 days post-germination, the phenotype became much more evident when plants reached, although on plates, later stages of development. Earlier, though, the phenotype is not really visible, indicating that this mutation has a role later in development. And at the subcellular level, we also found that the defects of IRA1 resides in the elongation zone, where the number and the size of the cells of this zone is reduced compared to wild type. So then we start manipulating the growth of Arabidopsis using sucrose, also using situations that induce the use of photosynthesis for the plant, in the sense that, for example, when we fed plants with sucrose, we didn't see the IRA1 root phenotype. However, in conditions that lead to the production of sugar, or the like continuous light, for example, or a night regime of light and dark, then we will see the short root phenotype of IRA1. So here there was a connection that we were not expecting. Now, hyperactivation of TOR leads to short root phenotype. And this is something that we established in our EVA1 story. So we then wanted to look at the phosphorylation of S6K in IRA1. And we found that in IRA1 root tips, the TOR was hyperactive, something that we could mildly suppress with the use of ACD, so the TOR inhibitor. So at that point, we treated IRA1 plants with the TOR inhibitor. And just by looking here at 12 days post-germination, you can see that in control plants where IRA1 double mutant has a root phenotype, by depleting the TOR activation, now we have that the roots can grow longer again. So I wanted to give you just a hint of where our lab is going to tell you that I found very fascinating how the TOR, which is so central to growth, and now we also understanding important for ER shape, actually has another controller that is IRA1. IRA1 wants to suppress a TOR activation. Indeed, when we don't have IRA1, TOR is hyperactive. And this makes sense because IRA1 is a controller of the UPR, so it really wants to prevent cells from producing misfolded proteins, while TOR, because it's a pro-growth factor, wants proteins to be synthesized. So there is this tug of war between IRA1 and TOR, in one trying to control the other, but mainly IRA1 trying to mitigate the action of TOR and make sure that TOR activity is such that the growth of the plant is not compromised by an excessive demand of proteins onto the endoplasmic reticulum. So with this, I would like to conclude my talk. I hope you appreciated the connection starting from an organelle to whole plant development as something that we find really fascinating in our lab these days. Thank you for your attention.
Thank you, Federica. That was marvelous. And back to my introductory remarks, I should have taken time to read a couple dozen papers from your lab in the last few years, because I learned a lot today. There should be time for some questions. If you want to ask your questions in the chat, or simply raise your hand or unmute and jump in, that would be great.
I have a question. So you talked about the three BCAAs the homeostasis of the three BCAAs can regulate the TOR activity. I was wondering for each individual one of the leucine, the leucine and the valine, do they contribute differently to the TOR activity?
So each of the chain amino acid causes the phenotype that I showed you. Whether one does it more than the other, we do not know. We were not able to quantify that. But what we know is that each of the three can stimulate TOR and cause the hyperbundling of actin.
Okay.
And all the other cellular phenotypes.
Because remember in the EVA1 mutant, you show the result that one of the amino acid has low, the content is low, and the other two has increased level.
Right, and then we use mutants of the other enzymes important for isoleucine production of valine production.
Okay. Yeah,
I'm very, very happy to share those lines if you need them.
Yeah, thank you.
Federica. Federica, let me jump in, if I may, with another question. It's about the ER plasma membrane contact sites where you provide nice cytological and protein-protein interaction data for clathrin-mediated endocytosis, and you show them coincident with each other but not overlapping directly, which I guess can be explained by the physical size of a clathrin-coated pit. Do you have evidence, or maybe I missed it, is there evidence that physiological cargo like FLS2 or BRY1 or some of the other ligand-induced uptake occurs at those sites?
Not published, so not that I know, Chris. I would expect the Aber Rosado at UBC maybe following that kind of study. We're not following that anymore. But I would expect yes, but
yeah, okay, no, that's cool. And the complementary question is, and I probably should know the literature, what is the evidence that ER plasma membrane contact sites are involved in exocytosis or are sites for secretion? Or are you currently thinking that it's just about uptake?
At the moment, we just think update, at least from the data that we have. And when we pull down VAP27, we didn't see anything forward, but for exocytosis, but it's possible we didn't catch it. But that's a good question, and we don't have evidence for anything.
Thanks, Federica.
Nick Carpita has a question.
As does Dr. Kessler, who I think was up first.
Yeah.
Go ahead, Sharon.
All right. Thanks, Federica. I really love watching those ER videos. It's really cool. I noticed in your chloroplast study, you had two proteins that were called LURs.
Yes.
Are those the same kind of cysteine-rich peptides that are pollen tube attractants that are called LURs, or is this a different type of protein?
So there is Luciana Reina is the postdoc that identified them. So Lure is just Luciana Reina, is the two words.
Oh, okay. So are they some kind of membrane proteins, or what are they?
Yeah. So they are type 2 membrane proteins. The classical, having a short transmembrane domain and a huge bulky cytosolic domain. And at the moment, what we have is this fascinating distribution. You really see these half moons, and we thought they were just the product of overexpression. And then we have complementary lines that still have this half moon distribution. So we're very excited about it, and hopefully you're gonna do something about it.
Thanks.
Welcome.
Hi, Federica. A long distance call in here. As far as export, I was thinking about the ER perhaps in vascular development and lignin formation. It's thought that the ER is actually the site of synthesis of the monolignals, but I wondered if that's one example where these aromatics could be shown at specific locations within the wall.
That would be a nice thing to test. Yeah, I mean, if somebody has the patience to do it, yeah. Chris, maybe you want to do it.
No, it's beyond me. I do have a question about the ER and actin connections, and I was thinking immediately about the trafficking of Golgi along actin and the stop and go mechanisms of Shalen, where the ER being, migrating with the Golgi might be at those spots. And I just wondered if anyone has ever looked at Golgi specific markers. I know there aren't such things, but Golgi concentrated markers versus ER concentrated markers, whether those stop and go kind of transitions are places where ER proteins are loaded.
So the reason why I'm smiling is that a couple of decades ago, there was such a hot topic that, it got a kind of resolved by saying that the Golgi seems to be continuously taking cargo from the endoplasmic reticulum. So it doesn't have to go to stop regions to on. And this is beautiful work from Akiko Nakano using super resolution microscopy. So there was this controversy over whether, yes, do the Golgi have to stop to collect cargo and then they move to the next station? But that doesn't seem to be the case. And I think the issue's been settled 3 years ago by Akiko's group, demonstrated that things can flow all the times to the Golgi.
But there would be an expectation then that the ER is used with the Golgi and migrating along the actin.
Which seems to be the case on Chris's data, that you can grab a Golgi and move it along. The question is, are all the Golgi attached? That's I think the question that we need to address.
All right, thank you. See you in a few weeks, I guess.
Dr. Chen.
I have a question about your last slides about the IRE1A, 1B double mutant. They showing a reduced root growth, and then if you add the inhibitor of the TOR, you can rescue the phenotype. You already know that if you add an inhibitor to the TOR, you induce the autophagy. So could that be maybe your double mutant, IRE1A, 1B, maybe you have some sort of physiological ER stress. You're accumulating some unfolded protein in the ER, and then when you're inhibiting the TOR, you induce some other protein degradation pathway, such as autophagy, that somehow can, which is now actually can target the ER protein, and therefore you can mitigate or reduce the ER stress, and therefore rescue the growth phenotype. Could that maybe as simple as that? Could it be?
I think it's as simple as that, as you're putting it. The point I think that I find fascinating is that TOR wants the plants to grow, right? And IRE1 really wants to monitor TOR activity, because otherwise there's going to be too much party for the endoplasmic reticulum, too much protein being produced. So I think we still do not know, and I think that's the biggest question, what is the connection between IRE1 and TOR, right? So the molecular connection is not there yet. I think autophagy is a downstream process. So while I agree with you that IRE1 has physiological UPR up because it's defective, right? And maybe some autophagy going on, I think we're still missing the molecular connection with TOR. So we only know that these two pathways are monitoring each other, but the connection is still missing. So that should be the next EMS screen. Thank you.
I have a question. Thank you for the beautiful videos. It was a very inspiring talk. I have a question that relates with something that you said on the beginning about homotypic and heterotypic interactions between the ER and the different organelles. And you mentioned there a fusogen that is responsible for fusing ER. So since you have made so many EMS screens, have you ever found any mutants that actually disrupts the interactions completely from the ER with some type of organelles? Or maybe because they were not found, maybe because they have little phenotypes?
It's a good question. So, but let me step just back a second. So when we do our screens, we use one marker. So a marker for the ER or the Golgi or the vacuole. We don't do screens with two markers for two different organelles. So I wouldn't, so we never looked for a mutant that would have separate organelles just because we didn't have the tools. To go back to your second part of the question, I think it would be lethal, to be honest with you. I think now having these connections that they seem to be so strong, I think they have, a physiological.
Probably lethal. Okay, thank you.
Any more questions? I know I have a whole pocket full, but I'll leave them for another occasion. Maybe we'll see you in a few weeks. No further questions. Let's thank Federica again for an amazing talk.
Thank you so much. It's been really my privilege to talk with you guys. And thank you for the questions. With this COVID going back to some kind of normalcy is just beautiful. So thank you.
Next time, I hope we see you here in person.
Absolutely. Take care. Bye-bye, everyone.
Bye.
Buddy.
Hey, bye-bye.
Hey, Federica. Oh, she's gone.